Good morning, everybody. Thank you for coming to this special GRC Connects event for the Synaptic transmission community. I'm very happy to bring three speakers to you today. Fan Wang is our first speaker, and her presentation will be followed by two short talks from Alicia Perkey and Bruce Herring. So let me introduce Fan. Fan is a molecular neurobiologist who has taken her skills and expertise in synaptic function to investigate the circuit basis for a number of interesting touch and pain-related circuits. She began her career at Duke University, where she carried out important research looking at the development of neural circuits, and more recently has been developing new tools for investigating cellular circuits involved in specific behaviors using immediate early gene expression and intersectional viral transduction. She has done important work elucidating the mechanisms for general anesthesia that appear to be important for controlling pain. And she has recently moved to MIT earlier this year, where she has set up her lab. And I'm sure she's looking for new students and postdocs. So trainees pay attention. So Fan is going to be talking to us today about the emergence of a rhythm with recurrent inhibition. And it's my pleasure to introduce Fan Wang to you.
Thank you, Allison. Should I just go? That's very flattering. I don't consider myself a synapse person, so I feel like I'm a little bit fake coming to the synaptic transmission field. And really happy to be here and hope to get all the feedbacks from the work. So you all see my screens? Good? Okay. So now I'm gonna talk about, this is unpublished, so hopefully to get lots of feedback from you. So life is a rhythm. All animals have rhythmic movement, and you'll see the video clips. I'll give you a few examples. Usually this kind of movement, rhythmic type of movement, is considered generated centrally. So there are names of the central circuit that generates such rhythmic movement. We call them central pattern generator, or central rhythm generator, central oscillator, so you name it. So I'm gonna call them oscillator just because it's the simplest way. So how do we know we find oscillator neurons? Yeah, so this is how my lab dress is on work now that we moved to MIT. Just kidding. So a oscillator generates movement in the absence of sensory input. To know the identity of oscillator neurons and mechanism of rhythm generation, so the examples are really sort of demonstrating in invertebrate that they did causal manipulation of specific cells that disrupt the rhythm, and there is recordings, preferably in vivo, in awake behaving animal, to show the cells are indeed fire rhythmically, and to identify either the intrinsic properties or the connectivity and circuit that generate such rhythms. So these three criteria, to be honest, have now been met to demonstrate a clear mammalian central pattern generator or oscillator. Mainly because it's really hard to do in vivo recordings in awake behaving animals in these oscillator neurons. Okay, so I'm gonna study this question and tell you the story about how we identify the whisking oscillator. Okay, so rodents use this back and forth whisker movement to explore their environment, especially in dark. In works several years ago by Kleinfeld and his colleagues, they find a region in the caudal hindbrain. Okay, so the region, when they did the electrical lesion, on the lesion side, what you can see is this back and forth rhythmic movement disappears of the whiskers, whereas the contralateral side, the whisking, this rhythmic movement is intact. So here in this hole, that's where they say this is where the whisking oscillator is. So they call this region VERT, ventral intermediate reticular zone. Okay, VERT is where the whisking oscillator is. But what is the molecular and cellular identities of the whisking oscillator neurons? That remains unknown. So we decided to take on this question. Before I'm going into that, just a little bit of background. So whiskers are driven by muscles surrounding them. It's like sling-like muscles. And then the facial motor neurons coming from the brainstem innervate these muscles and to protract and relax, and then the whisker retracts. Okay, so the simple hypothesis is that the whisking oscillator neurons are premotor neurons. Right. So they generate a rhythmic output such that the motor neurons will be rhythmically output and to control the muscles. So I'm going to tell you work done by two talented scientists in my lab, Jun Takato and Vincent Prevosto. So first we need the way to find the premotor neuron, where they are, and whether or not the VERT region contains premotor neurons. So we recently developed a strategy that allows us to trace adult premotor neurons. Previously, the premotor tracing using rabies virus was only done in neonatal mice. So we use a retrograde Cree in young mice that is still efficient enough to retrogradely infect the motor neurons in young mice. So this is done at P17. Then we inject the rabies complement into this region. So these motor neurons are now competent to complement the rabies. Then we inject the pseudotype of the rabies to only infect these motor neurons, and they will then jump to label all the premotor neurons. This works very nicely. I'm showing you 3 examples, 3 mice, and then the merged images, and you can see very reproducible the patterns of a whisker premotor neurons. Sure enough, we labeled premotor neurons in this caudal hindbrain region, VERT. Okay, so then the question is, what are the molecular identities? We took a candidate approach, and then interestingly, one of the candidates we tested, parvalbumin, co-localized with about half of the VERT neurons. I mean, most of your work with interneurons know parvalbumin. Here, parvalbumin, I mean, it's probably labeled many different types of neurons. So at least half of the VERT neurons that are premotor neurons for whisker motor neurons express parvalbumin. So how do we then specific these parvalbumin expressing VERT premotor neurons? So we made a strategy. It's a split Cree strategy. So Cree is split into 2 halves. And 1 half is knocked into the parvalbumin locus. So we generate a PV Korean animal. The other half is injected using a retrograde lentivirus into the facial motor nucleus. So that this retrograde virus will bring the other half of the Cree to many of the premotor neurons. Then only in parvalbumin positive premotor neurons, full Cree is reconstituted. And then if we specifically inject a Cree-dependent virus only in this region, then that would allow us to label a specific type of parvalbumin expressing premotor neurons. So we did this. So you can see green here is labeled parvalbumin positive premotor neurons. And then we did in situ hybridization. Shown here is a glycine transporter. And you can see almost all of them are glycinergic. And in fact, they're all GABAergic as well. I'm not showing you the data. So they're inhibitory cells, and keep that in mind. Okay, so we call these now VERT PV, premotor neurons. So I'm going to use this term a lot. So do they affect rhythmic movement of the whiskers. The first thing we did is express tetanus toxin using the same split Cree strategy, expressing tetanus toxin. We labeled about 200 neurons. Okay, so these neurons will be silenced with tetanus toxin, and this would be the silent side. This would be the control side. And you can see the control side still does this beautiful rhythmic movement, whereas the silent side, the whiskers stop, right? So this is just a heat map projection to show you the rhythmic whisking versus this minimum movement. And interestingly, they are in a very protracted, the very protracted region, okay? So, and the remaining small movement seems to have a low frequency, and usually the whole orofacial head region have some sort of connection with a breathing oscillator. So this is essentially the breathing rhythm. So this then led the conclusion. So this protraction without whisking leads to this model of how whisking is generated, which is consistent with previous finding that there is a tonic excitation that protracts the whiskers, and then you need rhythmic inhibition to relax the whiskers. And that's how you generate rhythmic whisking. And remember, our neurons, PV neurons, are actually inhibitory. So then the question is, are they firing rhythmically in vivo? And if they are rhythmic firing in vivo, do they fire in the retraction phase, right? Because you need them to be generating retractions. So to solve this issue, really we need an in vivo recording. So to record from identified PV VERD PV neurons. So this is a technique we use and now commonly used in neuroscience, except it's very hard to do it in brainstem, in a caudal hindbrain. So we, using the same split Cree strategy, express channel rhodopsin in the VERD neurons. And then we needed to then, so they would then respond to light. That allow us to identify them, that the neurons respond to light are the VERD PV neurons. Then we will see their in vivo pattern. So this actually took us 4 years, many mice. Eventually, we recorded 7 photo-tagged VERD PV cells from 6 mice. So the reason this is so hard, as you can see from post hoc histology, this is the electrode track, right? So the electrode is already pretty big, even though we use the silicon probe. It can easily lesion the VERD region. Now, initially, we have the optic fiber coming down here as well. So almost inevitably, the optic fiber lesion the VERD region. So we couldn't find any cells. Whereas we can blindly find rhythmic cells, but we don't know whether they're, respond to light. So in the end, we solved this question, this problem, by putting the light in the facial nucleus to using light to shine on the axon terminal to generate an antidromic spike that allow us to identify the VERD cells projecting to the facial nucleus. So here you go. You can see this is every 100 millisecond, we give a 10 millisecond pulse and we can induce spikes. So this is a phototag neuron. And I'm gonna show you a video. So this showing you whiskers, and then this is the protraction. So this is maximum protraction, and this would be the maximum retraction, okay? And then you can hear, I hope you can hear, but if you don't hear the those spikes, you can follow the red dots. That's sort of a duration of the spike, the burst. And that is summarized here. And you can see the spikes of the phototagged cells. 5 out of 7 have rhythmic bursting, and they are bursting in the end of the protraction phase and the beginning of the retraction phase. And that is plotted here. So the peak is at the beginning of the retraction. So that's nice, right? And that's, they fire rhythmically and they fire in the retraction phase. So here's a summary of all the rhythmic cells we recorded, they're actually separated into two groups. So these colored dots ones are mostly fire. They tune to the protraction phase. And then the black dots are the ones tuned to the retraction phase. And remarkably, all 5 phototag VERT-PV neurons fire in this retraction phase in the beginning of the retraction. There were 2 other phototag neurons that I'm now showing you. They have a slower rhythm. They seem to be more like breathing-like neurons. Okay, so that's good. And we're very happy that these cells fire as you would expect from the sort of oscillator neurons, right? Not only they're rhythmic, but also they fire in the right phase of the oscillation. So the beauty of having the capability of recording such cells in vivo in awake whisking mice is that we can see what are their firing patterns before the mouse whisks their whiskers, right? So I had thought, by all means, that they would be quiet. And then when the mouse started whisking, there will be boom, 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 rhythmically whisk. But that's not the case. It turns out they have tonic firing patterns, right? There just seems to be, in fact, even the protraction unit also have this sort of a tonic pattern, non-rhythmic tonic firing. Rhythm appears when the mouse decides to whisk. So this is also plotted here with this power spectrogram, and you can see no power in any frequency, and then the power emerges upon whisking, right? So this means rhythm generation is not an intrinsic property of these VERT-PV neurons. It's a network property, and that somehow through this top-down command that the mouse decides I'm gonna whisk, then the rhythm appears. So to begin to understand the network property that generates this rhythm, obviously we need to know the circuit. So again, we turn to transsynaptic rabies-mediated tracing. So we express using this, again, this viral strategy, split Cre, we express a rabies complement in the VERT neurons. Then we inject rabies to jump. And the VERT cells receive a lot of input throughout the brainstem, as well as top-down input from descending regions. So it's really interesting to see there's extensive local connections within the VERT, right? We label a lot of cells locally. And also we label cells from the pre-Boltzinger complex, which is a sort of a breathing center, breathing inspiration rhythm generator region. So to further sort of validate that VERT neurons connect to each other, what we did is we expressed, again, use the split Cre strategy in these VERT-PV cells, a membrane GFP and synaptic ruby. So the neurons will have GFP, and then their synaptic terminals will have red fluorescence from the ruby. And you can see when we label these cells, they project to the lateral part of the facial nucleus where the whisking motor neurons are, but they also sense ton of local botons in the VERT region. So these two are examples of the GFP positive cells, which are VERT-PV neurons to begin with. They receive the botons from other VERT-PV cells, right? So remember, these neurons are inhibitory neurons. So this suggests this region forms a recurrent inhibitory network. So here is basically a schematic summary of all the input. There's a recurrent inhibition, and there are other sources of inhibition from pre-Boltzinger complex and another region. And the top down from superior colliculus and cerebellum and red nucleus are excitatory input to these regions, and the mixed input from other reticular regions. Okay, so we wonder whether or not in this inhibitory network, it sort of resembles this, the half-center oscillators, which are all inhibitory. So there may be this recurrent inhibition plays a role in rhythm generation. To test this, we don't have a perfect way to test. So we have a sort of a proxy to test this hypothesis. This is done by, again, use the split tree strategy to express GFE3. Okay, so what is GFE3? GFE3 is a construct developed by Don Arnold's lab, which is essentially bringing a E3 ubiquitin ligase to degrade geoffrin, such that the post-synapse, inhibitory post-synapses are abolished. So as you can see, when we express GFP tagged GFE3 in these VERT-PV neurons, and we counted the puncta of geoffrin, they're drastically reduced. So we essentially compromised inhibitory synapses onto these PV premotor neurons, VERT-PV cells. Okay, so we call them VERT-PV GFE3 mice. Then we'll see what happens to whisking rhythm. So when we block inhibitory input onto these neurons. So the green is the GFE3 mice and black is control mice. Ignore the gray color, just focus on the green and black trace. And you can see this is rhythmic whisking. And the green is the whisking patterns in the GFE3 mice. So clearly they look irregular to you, so we lost this regular rhythmic oscillation. And the irregularity can be computed using something called transition matrix. So if it's a perfect sinusoid pattern, what you get is a elliptical pattern, right? If you add noise, then you start to have patterns like this. So when we computed the transition matrix of control and GFE3 mice, what we get is like this. So control mice clearly shows some sort of regularity in their rhythm, and that's regularity disrupted, suggesting rhythm is disrupted. One inhibitory input, two PV VERT neurons are blocked. So that's just nice. That says inhibition, synaptic inhibition is critical for rhythm generation. And the remaining movement, and you can see there's still some movement, and that actually looks like entrained by breathing. So this can be calculated by doing coherence analysis between the whisking in control and then the whisking in GFE3 mice. And you can see GFE3 mice, the remaining whisking are actually entrained by the breathing movement. So breathing traces are gray here. So that's quantified here. So there's a reduced amplitude, small amplitude of whisking, and then that is entrained by whisking. Okay, so now I'm gonna bring everything together. So normally in resting state, these cells fire tonically, but because there's no top-down excitatory input, the whiskers are just in a relaxed mode. There's no rhythmic whisking. There's a little bit of, back and forth movement, and that's entrained by breathing because that's how head regions have this movement entrained by breathing. Now, upon transition to normal whisking, the top-down excitatory input not only provide a tonic drive to the motor neurons to protract them, but also provide tonic excitation to drive this recurrent inhibitory network such that their inhibitory output is also strengthened and thereby, switching the network from tonic firing to rhythmic bursting. Okay, and this rhythmic bursting together with the tonic input to the motor neurons and generate the high frequency rhythm of whisking that you see. Now, when we use tetanus toxin to completely block the output of this network, even though they themselves probably are getting excited, but they cannot have any output to the motor neurons. So the net result is really no whisking, but a protracted position, right? In in terms of, in the case of when we block only the in synaptic inhibition within this network, but now blocking their output. So what happens is there's no rhythm because you need inhibition to generate the rhythm. So the end result is a stronger tonic output, but asynchronous. So what you do is you don't have the high frequency whisking, but because the stronger output, you have smaller amplitude, and therefore you don't have this protracted position because the retraction, the inhibition sort of counteract this tonic excitation. So you have this in this position, the smaller amplitude of whisking, and this amplitude of whisking is actually purely driven by breathing oscillators. So that's how we think the whisking oscillator works. So that's it. I hope I haven't exceed my 25 minutes time. This is my lab in MIT. Obviously, we are still social isolating. Mask is still required, and these are my funding source. We are actively looking for postdocs. Actually, so what I don't have data is actually Synaptic really going in, cut a brainstem slice, do a dual patch recording to show how these things work. So we're really looking forward to have someone who has a synaptic slice physiology experience to join our team at MIT. And thank you very much.
Thank you, Fan. That was great. We have a few questions that are just coming in. Maybe I could start off with a question about one of the tools that you have. This GFE3 ubiquitin ligase to disrupt inhibitory synapses by destabilizing gafferin is very interesting. How complete do you think that reduction in inhibition is? So do you think it's fully penetrant and you get rid of all inhibitory synapses, or is it about half of them? What's your assessment?
Counting the puncta result in a sort of, so we left with 20% gafferin puncta. So I would say a 5-fold reduction. And in Dan Arnold's paper, what they showed is you block basic synaptic inhibition, but you do not block the sort of extrasynaptic or perisynaptic spillover or other type of GABA. So it's really the phasic component is blocked. So yeah, it works really brilliantly. I usually very suspicious when, buy anything from magazine. This one works brilliantly. I highly recommend it.
We have a question from our group here about what the greatest challenge in the work that you hope to address when you move forward. I have to say it's quite impressive, all the challenges you've overcome. So, 6 cells over 4 years is like a, testament to your persistence. What's the next step for you?
Yeah, so it's actually really, we find a cell, right? And we know in vivo they oscillate, and then we think synaptic inhibition plays a key role. It's quite satisfying because in the breathing field, the pre-Boltinger is inspiration oscillator, but exactly what cell is the oscillating cell still remain controversial, right? And there is also a network property, but it's shown it's actually the excitatory input, maybe the rhythm generation. However, also you can cut a slice in neonates and they have breathing rhythm. You can cut a slice in adult because the whisking rhythm only emerges in sort of adultish, and by that time the brainstem slice is really hard to cut. So we have a 2-photon now. We're thinking of doing 2-photon guided patch clamp recording. That would be very challenging, but we're gonna give it a try. So
in,
if some
in the brainstem.
in the brainstem, yeah.
Hmm.
We'll see, yeah. But I mean, we're having a hard time cutting a nice brainstem slides in adult. Though all that myelin will just make the slides uh, ridiculous. So we haven't solved that. That's really the synaptic mechanism. Uh, the other thing, obviously, we're doing RNA-seq, right? So we're trying to find ion channels, and then we can do knockdowns, and then really drill at the mechanism. So we're still working on these things.
We have a few other questions. Let's see if we can address a couple more of them. Are any of the other functions of whiskers affected when you inhibit these premotor neurons, like exploration or texture discrimination?
So we usually only do one side. So we haven't done both sides. So the mouse, if you look at them in the cage, they kind of just use this side to manipulate this. Yeah. But we haven't really done inhibiting both sides. But it's so interesting in the cage. The side that touches the wall, and then we just use this side. Yeah. Yes. Good question.
Do the VERT cells express any other inhibitory cell markers?
So we haven't, that's what I'm saying. We don't have a typical RNA-seq data inside. So they do express things like pink one, the enkephalin. I don't know why. So they don't express somato7.
I see. Okay.
Yeah.
And then maybe one last question. Does the GFE3 disrupt Geffen effectively on the axon initial segment?
Good question. We haven't looked at that yet. So I don't know the answer.
Okay. Great. Well, I think everybody will have the chance to talk to Fan after we're done with this speakers this morning. So I encourage you to stick around. And when we go back into the open table format, you're welcome to just click on Fan's table. I think it's the first table, and you'll be able to continue the conversation with Fan.
Look forward.
Thank you so much, Fan. That was a great talk. I'd like to introduce our next speaker, Alicia Perkey from Duke University, where she's a postdoc in Scott Soderling's lab. Alicia was a graduate student with Mark Delacqua, where she was interested in glutamatergic synapses and their regulation using phosphorylation or palmitylation and other intracellular pathways. As a postdoc, she's shifted her focus to inhibitory synapses, which is a harder question because there aren't as many of them. But she's going to tell us about chemogenetic discovery of inhibitory synapse protein phosphorylation in vivo. Thank you so much, Alicia, for presenting your work.
Awesome. All right. Thanks, Allison. All right. I'm really excited to tell you about my project I've been working on Scott Soderling's lab for the past 2 years. And I don't think I need to tell anyone that phosphorylation has been found to be a fundamental mechanism that underlies many processes in our cells. And a lot of kind of advances in mass spectrometry have led to our ability to discern a lot of neuronal phosphorylation. And so here I'm just showing the excitatory synapse and a lot of the phosphoproteins there. And our understanding of phosphorylation really changed the way we think about plasticity at excitatory synapses. But this has been quite a challenge at inhibitory synapses just due to the fact that we can't biochemically isolate the inhibitory synapse. And so our lab within the past few years developed methods to actually look at inhibitory synapses and their proteins. So we can't even begin to understand phosphorylation at the inhibitory synapse without knowing kind of the parts list of the inhibitory synapse. So our lab and others have come up with this nice list. This is from our paper where we identified a number of proteins that are resident in inhibitory synapses. And among those were actually kinases and phosphatases. But as I said, it's been a challenge to actually do some of these kind of foundational phosphoproteomic studies in inhibitory synapses. But we know that phosphorylation is going to be important, and we, within the past few decades, have realized that actually just like excitatory synapses, inhibitory synapses undergo plasticity and these plasticity-dependent changes. So we know that there are likely kinases and substrates that underlie this behavior, but what are they? And so we have a couple of challenges when we think about phosphoproteomics and inhibitory synapses. So, and just in general, in addressing some questions. So one of them is, we want to understand phosphoproteomics at a cell type specific population, but what usually happens in these experiments is you pull samples and then you lose that cell type specific information. The next problem is we mix subcellular compartments. And so if you think about organelles within a cell, if you're putting that all together, you lose that kind of specificity. And so we want to get around these two issues in addressing our problems. And one way we can do that is using cell type specific markers, which then we can pull out only those cells in our phosphoproteomics experiments. And another way of doing this too is by using subcellular fractionation or labeling of these subcellular compartments. But no one has really combined these two. And so we thought, let's see if we can combine these for our phosphoproteomics of inhibitory synapses. And so the way that we do this is that we use a Cre-dependent turbo ID. So this is proximity biotinylation that we've been using, so in vivo bioID. And we get our subcellular specificity because we tag our turbo ID, this biotin ligase, to Geoffrin or colibistin. And the way this works is turbo ID will biotinylate nearby proteins within about 10 nanometers. And so when we tag this to our inhibitory proteins, we will then biotinylate inhibitory proteins and be able to pull them down using streptavidin. Then we digest these proteins to get peptides. And then from there, we can actually get our phospho-specific information by going over a resin that captures our phosphopeptides. So now we have this idea that we can have cell type specific, so whatever Cre line you want, turbo ID with a phosphoproteomics workflow. And so for my experiments, we had no idea if this was going to work, but I injected these turbo ID viruses into CAMK2 Cres so we could look at pyramidal cells. And then I injected 1 day of biotin for this turbo ID and then collected cortex and hippocampus. And what we found was that we actually could isolate phosphoproteins from inhibitory synapses in quite a few. And so the way that I've been looking at this data is through these kind of plots of proteins. So here I'm showing proteins and phosphosites of those proteins from both the colibistin and the Geoffrin or the Geoffrin baits. And so you can see this forms a dense, large interconnected network. And so a kind of a simpler way to look at this, I just wanted to see what kind of proteins I had here. So I use Go and domain analysis, and what I see is what you'd expect from an inhibitory synapse, which are postsynaptic and cell adhesion kind of proteins. I see kinases and phosphoproteins coming out in this analysis. And if we look at each individual kind of at the protein level, I see proteins that are classically known to be at inhibitory synapses like GABA A receptors and Geoffrin, but I also see cell adhesion molecules like the catenins and neuroligands. I also see kinases like pKa and CAMK2 known to be important in excitatory plasticity, and also some inhibitory plasticity models. And I see some other signaling molecules like the GTPases and GAPs. Okay, so looking at this data another way, I now am showing you these proteins. So the protein nodes are in gray, and then their phosphosites are in colors all around. The size correlates to the fold change, and then the actual coloration denotes the FDR. And so here I'm just showing you what's enriched in both colibistin and Geoffrin baits. And ways I've been trying to think about this data, because it is quite a bit of data, is what kind of motifs are coming out of this data. And so when I align all of these sequences from these individual peptides, I see a couple of motifs coming, jumping out. And so I use Momo to come up with these, to find the enriched motifs within my data. And what I see is these three that when I use TomTom and kind of go back in the literature to see what kind of kinases go along with this, I see again, pKa is coming out, casein kinase, GSK3 beta for this top, the middle, PKC, CAMK2, and the bottom you can see is very clearly proline directed phosphorylation. So we have these really nice pathways we can actually pursue in future in this data. Okay, so just as an example, I want to show you casein kinase. So using kinase substrate prediction, I looked at what kind of kinases are going to be phosphorylated in these sites. And casein kinase popped out here, and I just have those in yellow. And casein kinase is very interesting. It's been studied a lot in circadian rhythm, and there are a number of isoforms of this casein kinase, and it's been implicated in sleep disruption, and also it's been found to be upregulated in Alzheimer's disease and could be implicated in neurodegeneration. So I thought this would be a very interesting example to look into, and it hasn't been really studied at the inhibitory synapse as much, and it was in that original paper from our lab looking at in vivo bio-ID at inhibitory synapses. And so what I did is I just took a pharmacological approach. I induced inhibitory LTP using NMDA and CNQX for 2 minutes, let it recover, either in the absence or presence of a casein kinase inhibitor. And here I'm using the casein kinase 1 delta inhibitor because that is the one that has been implicated in a lot of rhythms and the Alzheimer's disease and neurodegenerative diseases. And so I then used patch clamp electrophysiology to measure inhibitory synaptic transmission. And what I see is that in the presence of ILTP in orange, you can see this nice increase in both frequency and amplitude of mini iPSCs, but that's actually blocked in the presence of this casein kinase 1 delta inhibitor. So this tells us that our data can inform us what's going on in the inhibitory synapse. And we have a lot of now really interesting kinases and substrates to pursue. And as casein kinase 1 is a really interesting example, we can get some really useful information out of this. So just in future, what I want to do is look at, again, now plasticity. So now I have this kind of interesting phosphoproteomic workflow where we can look at inhibitory synapses. So I want to look at the plasticity behavior of phosphorylation there. And then I also want to look at kinases. What are these kinases doing? Who are they interacting with at inhibitory synapses? All right, and with that, I thank you for your attention. I want to thank my lab, the Duke Proteomics Core, the organizers, and funding, and I'm happy to take any questions.
Thank you, Alicia. That was a great talk. We have time for just a few questions. Let me start off here. Alicia, you have a lot of candidate proteins. I think casein kinase 1 is a great potential target, but there are likely to be multiple substrates for that. So what's your best guess for this transduction pathway by which the amplitude of inhibitory synapses is augmented when you have excitatory synaptic potentiation?
Yeah, that's interesting. I think it's likely a GABA receptor mediated pathway. So I think that would be really interesting. And I'm really interested in receptor trafficking and modification. So I think that would be a good place to start. Casein kinases are very interesting because these particular kinases recognize pre-phosphorylated substrates. So I think that it will get a little murky there, but I think it's really interesting. So I think it'll be interesting to look at the interplay too of, are they recognizing already pre-phosphorylated pools of maybe a receptor and recruiting them or something like that?
Oh, really interesting.
Yeah.
We have another question from our audience here. Did you observe any region-to-region differences in the phosphocandidates?
Oh, no. So I haven't looked at region specificity, but I think that's super interesting. And so for my experiments so far, I've just been pooling cortex and hippocampus to get as much material as I can, because as you can imagine, phosphoproteomics requires a lot of material. So, but no, that's a great question. I'm so interested.
One of our last question is, what's the sort of spatial span of your turbo ID approach? Because, from at least our understanding, it looks like excitatory and inhibitory synapses can be quite close. Are you sure you're not sampling excitatory synapses at all? Did you pull up any excitatory synaptic proteins?
I did, and I actually don't think that's a contaminant because we know that there are dually innervated synapses, and I actually do see proteins that I know that are at dually innervated synapses. So I think we just need to be careful with the conclusions we draw from this, but I think that inhibitory synapses on spines are still valid inhibitory synapses.
Absolutely. Really exciting. Thank you, Alicia. We're going to move on to our last talk of the day from Bruce Herring. And Bruce Herring is a faculty member at USC, where he's been there since 2016. He studies glutamatergic synaptic regulation in disease and has done some really exciting work taking genomic data to develop really rational hypotheses to investigate genetic disorders and link them to synaptic functions. So Bruce, let you take it from here.
All right, thank you so much, Allison, for that introduction and for the opportunity to share some of our recent unpublished work with you all today. Thanks for being here. The title of my talk is Self-Specific Expression of SAP97 Links Augmented Glutamatergic Synapse Function to Schizophrenia. So this slide illustrates our current genetic understanding of schizophrenia. Each one of these dots represents a genetic perturbation that has reached whole genome significance for the disorder. For the purposes of this talk, I want to call your attention to this particular genetic perturbation, 3q29 deletion mutations. Individuals with, so these particular deletion mutations result in the largest increase in the risk of developing schizophrenia of any known genetic perturbation that's currently been identified. Individuals with these deletions within the third chromosome, this confers a greater than 40-fold increase in the risk of developing schizophrenia. 5 known genes and 17 uncharacterized transcripts are deleted. And these are the 5 known genes that a copy is deleted. Of these, DLG1 is the most promising candidate gene. The DLG1 gene encodes the protein SAP97. The reason why DLG1 is sort of the favorite is that there are individuals that have been identified with schizophrenia that harbor missense mutations, single amino acid substitution mutations that are clustered specifically within SAP97's PDZ2 domain. So what do we know about SAP97? SAP97 is a member of the McGuck protein family. Other members of this protein family include PSD95, PSD93, and SAP102. We know quite a bit about these members of the McGuck protein family. PSD95, PSD93, and SAP102 exist within the postsynaptic density of glutamatergic synapses. They act as scaffolding proteins that hold AMPA and NMDA receptors in close proximity to the presynaptic side of the glutamatergic synapse and allow them to be efficiently activated by presynaptically released glutamate. And when PSD95, PSD93, and SAP102 are eliminated in neurons, this causes AMPA and NMDA receptors to fall out of these synapses, resulting in reduced glutamatergic synapse function. SAP97, on the other hand, is a bit of an odd duck in the protein family. Previous attempts have been made to uncover a role for SAP97 in governing glutamatergic synapse function. A number of these studies have been conducted in CA1 pyramidal neurons of the hippocampus. CA3 pyramidal neurons send their axons out and synapse onto the dendrites of CA1 pyramidal neurons. These are Schaefer collateral synapses and are the most extensively studied glutamatergic synapses in the brain, are generally used as a model for all glutamatergic synapses in the brain. We found that when we knock down SAP97 expression in CA1 pyramidal neurons with an RNAi, this produces no effect on glutamatergic synapse function. Synaptic AMP receptor currents are unchanged. Synaptic NMDA receptor currents are unchanged. So like I said, this is data from our lab, but this has actually been shown previously by Roger Nichols' group, who knocked out SAP97 in CA1 pyramidal neurons, and so no effect on glutamatergic synapse function. And so this has led groups to conclude that deleting SAP97 has no effect on synaptic transmission. So this led us to wonder if we've been looking in the right place. So given that SAP97 is implicated in schizophrenia, we went to the literature to identify regions of the brain that are implicated in schizophrenia and found growing literature suggesting dysfunction within the dentate gyrus may play a role in the development of symptoms associated with schizophrenia. This prompted us to look at where SAP97 is actually expressed in the hippocampus. And what we found was pretty exciting. So this is immunolabeling of SAP97 protein in a rat hippocampal slice. We find that SAP97 is expressed in CA1 pyramidal neurons, but its expression and localization is restricted to the cell bodies of these neurons and is absent, as far as we can tell, in the dendrites of CA1 pyramidal neurons, which is where the shapeful collateral synapses actually are. But in the dentate gyrus, we see something very, very different. In the dentate granule neurons of the dentate gyrus, we see that their axon or their dendrites are chock full of SAP97. And so we were very interested in whether SAP97 may be regulating glutamatergic synapse function at synapses that occur between the axons of entorhinal cortical neurons that synapse onto the dendrites of dentate granule neurons. So as I showed you before, when we knock down SAP97 and CA1 pyramidal neurons, this has no effect on glutamatergic synapse function. We then went and knocked down SAP97 in dentate granule neurons, and what we found was very, very different. As opposed to nothing or no effect that we saw in CA1 pyramidal neurons, in dentate granule neurons, when we knock down SAP97, we see a massive increase in synaptic AMP receptor current amplitude. There's about a 4-fold increase in the amplitude of these AMP receptor mediated currents. So this is consistent with some previous work that's been done looking at characterizing the subsynaptic localization of SAP97. This is a glutamatergic electron micrograph of a glutamatergic synapse. And in this case, the postsynaptic neuron was transfected and is expressing recombinant SAP97. And what was found was that SAP97, rather than going to the postsynaptic density where PSD95, PSD93, and SAP102 are, SAP97 seems to localize sort of in perisynaptic regions of glutamatergic synapses, sort of in this area. And it was proposed that this perisynaptic SAP97 may hold or sequester a pool of AMP receptors sort of outside of the postsynaptic density. And so in the event that we're reducing SAP97 expression as we are in dentate granule neurons, this would likely mobilize this pool of perisynaptic AMP receptors that are then allowed to reach the postsynaptic density and therefore increase the number of AMP receptors that you have at these synapses. So another very interesting thing about SAP97 is that it is the only McGuck protein that can bind directly to AMP receptor subunits. And so more specifically, the AMP receptor subunit GluA1 has a C-terminal PDZ binding domain that binds directly to SAP97's PDZ2 domain. This is a very well characterized interaction. The structure of this interaction has been solved. Another very interesting thing is that SAP97's PDC2 domain is where we see these clustered schizophrenia-related missense mutations. So while we were certainly interested in the impact these mutations would have on synaptic transmission in dentate granule neurons, we first wanted to ask what happens when you get rid of GluA1's PDZ binding domain. If this interaction is truly important or truly responsible for SAP97's synaptic role in dentate granule neurons, then you would predict that getting rid of GluA1's PDZ binding domain would also produce a big increase in synaptic AMP receptor current in dentate granule neurons. So we did molecular replacement experiments where endogenous GluA1 was replaced with GluA1 missing its PDZ binding domain. And we did this both in CA1 pyramidal neurons and in dentate granule neurons. When we molecularly replaced GluA1 with GluA1 missing its PDZ binding domain in CA1 pyramidal neurons, we found that this had no effect on synaptic AMP receptor current amplitude. This really wasn't that surprising. This has actually been shown previously by Rick Hugenier's lab. But when we did the same manipulation in dentate granule neurons, we see something very, very different. We see this very dramatic increase in synaptic AMPA receptor current in dentate granule neurons. So by eliminating this interaction between GLUA1 and SAP97, either by reducing SAP97 expression or by eliminating GLUA1's PDZ binding domain, it would seem that this results in a mobilization of a perisynaptic pool of AMPA receptors that can then result in an increase in synaptic AMPA receptor expression. So then, of course, we are interested in what impact the schizophrenia-related missense mutations in SAP97's PDZ2 domain have on glutamatergic synapse function in dentate granule neurons. So again, we took a molecular, we used a molecular replacement approach, showed you before that knocking down SAP97 expression in dentate granule neurons produces a large increase in synaptic AMP receptor current. We found that knocking down SAP97 and replacing endogenous SAP97 with recombinant wild type SAP97 restored normal glutamatergic synapse function in dentate granule neurons. But in stark contrast, when we replace endogenous SAP97 with these mutant forms of SAP97 that harbor these schizophrenia-related missense mutations, we see, again, these very large and likely pathological increases in synaptic AMP receptor current amplitude. And so in individuals that have reduced SAP97 expression, like what you would expect in individuals with 3q29 deletion mutations, or in individuals that harbor these schizophrenia-related missense mutations in SAP97's PDZ2 domain, we would predict that you'd see these large increases in synaptic AMP receptor function in dentate granule neurons of these individuals. So does SAP97 dysfunction in the dentate gyrus produce schizophrenia-related behavioral phenotypes? So we've started to try to answer this question in rats. Schizophrenia-related behavioral phenotypes or identifying schizophrenia-related behavioral phenotypes in rodents is a notoriously difficult task. We've decided to start by looking at effects on contextual episodic memory. There's a fairly large literature detailing contextual processing deficits in individuals with schizophrenia. And it has been proposed that deficits in contextual information processing may underlie the development of a number of the positive symptoms associated with schizophrenia. So what we've done is to locally knock down SAP97 expression in the dentate gyrus of rats using stereotaxic AAV injections of our SAP97 RNAi. We then looked at the performance of these animals on a novel object in context paradigm and find that inhibition of SAP97 in the dentate gyrus results in a pretty profound reduction in contextual information processing in these animals. We've also knocked down SAP97 expression in the CA1 region of the hippocampus, where we found that SAP97 plays no role in governing glutamatergic synapse function. And we found that this manipulation had no impact on contextual information processing in these animals. So from this, we conclude that SAP97 function specifically in the dentate gyrus is sufficient to produce schizophrenia-related behavioral phenotypes in rodents. And altogether, we think that we may have identified a specific synaptic regulatory pathway at a specific synapse in a specific brain region that, when disrupted, could very well contribute to the development of schizophrenia-related disorders. But obviously, we have quite a bit more to do. But with that, I want to thank my laboratory, in particular Uni K, who's done the bulk of this work, as well as our collaborators at the Milski Lab and the Catridge Lab, and who've helped with this study, as well as our funding sources. And so with any remaining time, I'm happy to answer questions.
Thank you, Bruce. So I'd just like to remind everybody that after this, we will have the chance to go into the Remo format, and you can talk to the speakers at their tables. I think we have time for one question here. So from Sam Young, we have a question. At what point are you knocking down SAP97, and how does this relate to disease conditions? So presumably in someone who's schizophrenic, they have this deficiency from early development. But at what time point are you performing this manipulation?
So in terms of the physiology, we are knocking down SAP97 in hippocampal slices from P6 animals, so relatively early in development. For the behavior, we've found empirically that getting robust learning on this novel object and context paradigm, older animals generally learn this behavior better when they're older. In this case, we're doing our AAV injections at around P70. So we are sampling different developmental ages, but certainly going forward, we are very interested in generating mice that harbor these missense mutations that are related to schizophrenia, and sort of tracking alterations in synaptic function and behavior as they progress sort of throughout development.
I think in the interests of equity, we should probably move people to these different tables now to continue the discussion. The GRC has allotted us about another 55 minutes for us to talk to each other. So I think the format is sort of formally till 12:30 Eastern time, but the tables and the site will be open until about 1 o'clock. And so I'd like to give people plenty of time to interact with each other. Bruce, it was a great talk. A lot of really interesting questions have arisen. And I know people are going to come and talk to you as we move on to the next format of this meeting. So thank you very much to all of our speakers. And I will see you then on the remote floor.